Alright, this tutorial is on chemical reactions in the human body, and we're going to begin this discussion with a little side step into a bit of physics so that we can understand what we mean by the word energy as we use it in a biology class. So we've got two different forms of possible energy, potential and kinetic energy. You can see them listed here. And so we want to go through each of these forms of energy and write down a definition, but then also really pay attention to how these definitions apply to the human body, which is somewhat different than you might have heard them described as applying to with general physics principles. So potential energy is usually defined as the energy of position or composition. So this is energy that is not doing work at the moment, it's just being sort of held. And so you might have at one point encountered the tried and true physics definition of potential energy of a ball at the top of a hill. So we'll go ahead and draw that in because it is sort of the classic example. So if you have a ball at the top of the hill, the ball is just sitting at the top of the hill, it's not going to go anywhere, but it does have some energy based upon both its position and composition. If we can push it a little bit, it's going to roll down this hill and we'll have motion. So we've got energy because it's at the top of the hill right here. In addition, there's an energy of composition because depending upon what this ball is made of, it's going to have more energy that it can expend as it rolls down the hill. You might think of a ball of marshmallow rolling down a hill versus a ball of lead. And if you're standing at the bottom of the hill, you'd much rather have that marshmallow hit you than a ball of lead. So there's a different amount of energy based upon what's inside that ball or what that ball is made up of. So that's a great definition, but it doesn't make any sense when we move inside the human body because I don't know about you, but I don't have balls sitting at the top of hills inside my body. But we do have other versions of energy of position or composition. So I want to give you the two big examples of potential energy in the human body. The first one is a concentration gradient. I'm going to draw that off to the side here. So if we have some sort of barrier, like, say a cell membrane, and on one side of the cell membrane we have a bunch of a certain type of molecule. And on the other side of the membrane we have very little of that molecule. You might remember from your intro biology class that substances always, if they're able to, move down their concentration gradients. And so right here, because we've got this barrier here, we've got a concentration gradient that's being held in position, and we have energy of position because we've positioned all of this molecule on this side and very little on this side. So we've got energy of position. A second form of potential energy that we'll utilize in the human body is the energy found within atomic bonds. So I'm going to write this as molecules because all molecules have atomic bonds. Let's go ahead and we'll just draw ourselves a little quick water molecule right here. And you can see I've drawn these two lines to indicate the bonds that exist in the water molecule, right? So there is energy available if we can break this bond or this bond. And so the fact that there is energy available upon bond breaking means that this molecule has an energy of composition because these bonds represent an ability to access energy. But not all of these bonds are created equal. So some bonds contain more energy than others. And so, for example, we're going to start talking about a bunch of molecules that have carbon backbones. Of course, these carbons are going to be bonded to other things since carbon will form a total of 4 bonds with the molecules, with the atoms around it. But these bonds here are quite energy rich, and so if we can break those, we are able to access the energy inside. So we've got energy of composition. This molecule here there's a lot more energy in these bonds than in these bonds right here. So that's potential energy, energy of position or composition, and the main two forms of potential energy we'll be playing around with here in class is concentration gradients and the energy found within every molecule, some of which have more than others. All right, so what's the definition of kinetic energy? Kinetic energy is the energy of motion. If we go back to our classic physics example of the ball, as we push that ball and it rolls down the hill, we've now got energy of motion. We've converted the potential energy of the ball's position to kinetic energy of the ball physically moving down the hill, energy of motion. Again, since we don't have balls on hills in our body, what does kinetic energy mean inside of our body? Well, we've got lots of examples of kinetic energy inside the human body, and one of them ties back to that form of potential energy we first discussed, which is a... So remember that I had trapped, in my first definition of a concentration gradient, a particular molecule on one side by the presence of this membrane here. But if I provide an opening in that membrane, some sort of little doorway through the membrane, then I have a place for these molecules to move down their concentration gradient, and of course the key word was that move. Where are these molecules going to go? They're going to start moving down their concentration gradient. And so we can describe one version of kinetic energy is molecules moving down a concentration gradient. And I'm using the word down, but the other word that you might have heard used that also works is with. So molecules moving with the concentration gradient. All right, a second type of kinetic energy that we'll find inside the human body, other than molecules moving down their concentration gradient, is molecules that actually have a component to them that can change shape. And the most typical molecule that would do this is a protein. So proteins changing shape. And we'll see this a lot this quarter, that we'll talk about a protein being triggered to change shape by a change in charge around it, or maybe by some other atom or molecule attaching to it. This is very typical of a type of job that a protein might have, is that as we change the environment around it, give it a particular molecule or ion, or change the charge, the protein physically changes shape. And one of the ones we'll see a lot is one that I actually kind of drew up here for you already, but I'm going to draw it again here. If I have a protein embedded in a membrane, so this is a cell membrane, sorry, it's awfully simple. And this is my protein sort of doorway, if you will. A lot of protein doorways will have a sort of closure on them. And if we change something about the environment around this protein, we can get this trapdoor to open, and now my doorway is available for something else to go through. So proteins changing shape are a very typical type of kinetic energy that we'll be employing a lot this quarter. All right, now that we've got these two terms out of the way, let's move on to talk about the word metabolism. The word metabolism refers to all of the reactions that are happening inside of the human body. I'm going to shorten reactions to this, so you'll see this shorthand from me. So all reactions in the human body. And this refers not only to the reactions that you might have thought of when I first said the word metabolism, actions of digestion, that sort of thing, but any event in the body that has one molecule being converted into another molecule, broken down, built up, all those sorts of things. So any reaction that occurs in the human body, we use the word metabolism to describe all of them. And these reactions are happening all the time. So this is a very frequent thing that occurs everywhere all the time as cells change molecules, build molecules, break molecules down. So it's helpful to sort of subset what kinds of metabolic reactions can you have. Well, basically you can have two types. You can have metabolic reactions that build things up, metabolic reactions that break things down. And so we use the two words anabolism and catabolism to describe that. So anabolic reactions, and we could also say anabolism. That's a kind of a funny word. It doesn't quite roll off the tongue. These are reactions that build larger molecules from smaller ones. And on the simplest form you can think of, if I've got these two small molecules, I might perform a reaction that bonds them together to create one larger molecule. That would be an anabolic reaction. And then the opposite, a catabolic reaction, or catabolism. These are reactions that break larger molecules into smaller pieces. And of course, if we have a big molecule here, we can perform a reaction on it and break it into two separate pieces. That would be a catabolic reaction. All right, so these terms of anabolism and catabolism are quite general terms to describe any reaction that builds things up and any reaction that breaks things down. But in the human body, these reactions get a little more specific. We can get a little more detailed in exactly how an anabolic reaction occurs, how a catabolic reaction occurs when we're building and breaking down a group of molecules called organic molecules. And so when we use these two terms in the human body, we actually get more specific with exactly how the reaction is happening, and so our terminology can get more specific. And so I'm going to write the terms in here, but then we'll go to a fresh page to describe them. So in the human body, the anabolic reactions that we use are specific kinds of reactions called dehydration synthesis. Again, I'll explain those on the next page. The catabolic reactions in the human body that break down organic molecules are called hydrolysis reactions. And these two terms are talking about specific types of anabolic and catabolic reactions. So we'll move on to a fresh page and describe dehydration synthesis and hydrolysis. Okay, so let's discuss dehydration synthesis and hydrolysis. So dehydration synthesis is an anabolic reaction. We are building a larger molecule from smaller molecules. This particular anabolic reaction is called dehydration synthesis because of the way in which a molecule of water is produced by this process. And so this word dehydration, we are removing some water over here, and it's getting produced by the equation, or the reaction, excuse me. And this word synthesis I find very helpful because sometimes these reactions are just called dehydration reactions, but I find that it helps me to remember that this is a building reaction if I plug that second word on there, dehydration synthesis, synthesis meaning to build. So let's see what we've got here. All right, on the left-hand side of the equation, my reactants, I've got two little molecules. And you can see I've been kind of simple. I haven't really shown you what atoms make up this molecule within this little bendy part, but you can see I have pointed out to you that hanging off of this end of my molecule is a hydrogen atom. And hanging off this end of the molecule, I have both an oxygen and a hydrogen molecule. This is called a hydroxide group. Another the same kind of molecule, a bunch of atoms in here with a hydrogen hanging off one end and a hydroxide group hanging off of the other. So we're going to use these two molecules and we're going to covalently bond them together into a single larger molecule right here. So we've got all these pieces all covalently bonded to each other. I'll come back to this enzyme that is facilitating this process in just a minute, but I want you to take a look at something that I have not accounted for yet in my equation. I had to remove some atoms off of the ends of both of these little molecules in order to find a place to covalently bond them together right here to form this covalent bond, which is strangely long. And so I had to remove these atoms here, and so I need to account for them. I need to show them being produced right here. And so I'm going to go ahead and add in the final piece of my equation here, and that is that I have produced a water molecule here. And I hope you can see that because I've removed this hydroxide group from one of my molecules and this hydrogen group from the other in order to find the place, I'll draw this here, then that produces a water molecule for me. And so at the end of the day, my dehydration synthesis reaction, as I combine smaller molecules to form a larger molecule, I produce a water molecule in the process. Water is produced or given off, hence our word dehydration. All right, let's look at hydrolysis, and then we'll come back to the presence of this enzyme that you can see is necessary for both reactions. So a hydrolysis reaction, of course, you know that a hydrolysis reaction is catabolic. We are breaking a larger molecule into smaller pieces, and I hope as you look at this reaction you can see that, well, it looks a little familiar. It looks like we've just taken the dehydration synthesis reaction and run it in the opposite direction, which is true, we have. So here, my reactant, I have a large molecule, and as I facilitate this process with an enzyme, I can break this covalent bond right here to produce two smaller molecules. Again, though, I haven't accounted for everything just yet, so let me grab my red pen here. You can see as I break this bond right here, I need to satisfy the octet rules where I broke that bond. And so I can do that by bringing a water molecule to the mix. So if a water molecule is available, then I can tack on the hydroxide on one molecule, and the hydrogen on the other, and I've provided the proper atoms in order to satisfy the octet rules of this small molecule and the octet rules of this small molecule. So hydrolysis reaction, I'm breaking a covalent bond of a larger molecule and using a molecule of water to satisfy the openings I've created by breaking that covalent bond. Okay, so now let's go to the enzymes and what their purpose is in each of these processes. So these processes will happen on their own, but they happen on their own very slowly. And in a living body, we need things to happen quickly. We need to be able to break down the food we're eating quickly. We need to get energy out of molecules quickly, and we need to build the molecules that we need to survive quickly. So we need these reactions to happen much faster. And so an enzyme is a protein that makes these reactions happen much more quickly. So let's get a few words in here. So enzymes are proteins that catalyze, that means make happen faster, reactions. We need these enzymes in our body. We'll have different enzymes for each reaction. So this enzyme here, different one than this enzyme here to catalyze this other reaction. So each enzyme is available to catalyze one specific reaction, and then those reactions can happen much more quickly when the enzyme's available. One final little chunk of information about enzymes, because we're going to be using them a lot this quarter. We're going to be noting how they facilitate so many reactions in the human body. But enzymes work at different speeds based upon different parameters of what's going on. And one of the main things that can influence how quickly a reaction can happen is what's the temperature. So in general, we'd say that the warmer the area, the faster the reactions happen, up to a point. So warmer equals faster until it gets so warm that my enzyme actually falls apart. And if the molecule can't hold together, it can't catalyze the reaction until the enzyme, I'm going to use this word denature because that's what I am really talking about when I say the enzyme falls apart, till enzyme denatures. And in the human body, that's at about 104 degrees Fahrenheit, and of course right now I've forgotten the Celsius. I'll have to look that up. So up to about that temperature, enzymes will work faster and faster and faster. And so a little bit of heat when you've got a fever because you're sick, part of that is helping your own body's defense systems work a little faster. Their enzymes work faster. They can battle that pathogen a little faster. But if you get too hot, things don't work so well anymore. All right, so temperature up to a point. All right, another thing that can change how quickly an enzyme can work is how much, I'm going to put concentration, how much reactant do you have? Remember, the reactant is the thing that the enzyme is working on. In hydrolysis, this was the reactant. In dehydration and synthesis, these were the reactants. And I'm sure this isn't very complicated for you to see, that if I have more reactant, then I can make more reaction happen. The enzymes have something to work on. So concentration of reactant is just going to speed up a reaction because then we've got more material to work with. And then finally, we've already described the fact that without enzymes at all, then these reactions are going to happen very slowly. So how much catalyst? Again, I use the word catalyze right here, right? The catalyst is the enzyme. So how much enzyme do you have? If you've got more enzyme, you can make more reaction happen, as long, of course, as you've got reactant available. So those are things that can drive how quickly reactions can happen. What's the temperature? How much substrate do you have to work with? How much reactant do you have to work with? And how much enzyme do you have to work with? Okay, and to wrap this up, I just want to point out to you that these processes of dehydration, synthesis, and hydrolysis, we're going to keep coming back to them all quarter long. So make sure that these concepts make sense. Make sure that you feel comfortable being able to draw out these reactions and describe what's happening in them, accounting for everything in the reaction. So for example, in dehydration synthesis, you'll tell me you start with small pieces. You're going to want to tell me that we've got hydroxide and hydrogen pieces hanging off the ends. We need an enzyme to facilitate. We come up with a larger molecule, and we've produced a molecule of water in the process. And then, of course, hydrolysis, basically the opposite. What do we start with? We have a reactant, which is a large molecule, requires a water molecule to input, and as that enzyme puts stress on this bond, the water molecule can break apart and fit into those locations and satisfy those, and we have two small pieces as a product.